Organization of the cardiovascular system. The function of the circulation is to serve the needs of the body tissues, to transport nutrients to the body tissues, to transport waste products away, to transport hormones from one part of the body to another, and in general to maintain an appropriate environment in all the tissue fluids of the body for survival and optimal function of the cells. The rate of blood flow through many tissues is controlled mainly in response to their need for nutrients. In some organs, such as kidneys, the circulation serves additional functions. Blood flow to the kidneys, for example, is far in excess its metabolic requirements and is related to the excretory function, which require that a large volume of blood is filtered each minute. The heart and blood vessels in turn are controlled to provide the necessary cardiac output and arterial pressure to cause the needed tissue blood flow. What are the mechanisms for controlling blood volume and blood flow, and how does this process relate to the other functions of the circulation? These are some of the topics and questions that we discuss in this section on the circulation. So, physical characteristics of the circulation. The circulation is divided into systemic circulation and pulmonary circulation. Systemic circulation and pulmonary circulation, because the systemic circulation supplies blood flow to all the tissues of the body except the lungs, and so it is called greater circulation or peripheral circulation. It is also clear from the lecture that the heart is in fact two pumps in series. The right heart pumps blood into the pulmonary circulation while the left heart pumps blood into the systemic circulation. Functional parts of the circulation. Before discussing the details of circulatory function, it is important to understand the roles of each part of the circulation. The function of the arteries is to transport blood under high pressure to the tissues. For this reason, the arteries have long vascular walls and blood flows at a high velocity in the arteries. The arterioles are the last small branches of the arterial system. We learned two types, like pulmonary circulation and systemic circulation. Okay. Next, functional parts of the circulation. The function of the arteries is to transport blood under high pressure to the tissues. For this reason, the arteries are strong vascular walls and blood flows in a high velocity in the arteries. Then the arterioles are the last small branches of the arterial system. Arterioles. They act as control conducts through which blood is released into the capillaries. Arterioles have strong muscular walls that can close the arterioles completely or can by relaxing dilate the vessels several fold, thus having the capillarity of vastly altering blood flow in each tissue in response to its needs. These blood vessels are sometimes called resistance vessels. When we try to learn about the distribution of blood in percentage of total blood in the different parts of circulatory system, we could see pulmonary circulation 9%, systemic circulation 84%, superior vena cava, aorta, heart about 7%, inferior vena cava, then systemic vessels, arteries 13%, arterioles and capillaries 7%, veins, venules and venous sinuses 64%. Remember, systemic circulation 84% and pulmonary circulation 9%. The function of the capillaries is to exchange fluid, nutrients, electrolytes, hormones and other substances between the blood and the interstitial fluid. They are therefore sometimes called exchange vessels. To serve the role, the capillary walls are thin and have numerous minute capillary pores. Capillary pores. Capillaries and capillary pores. Permeable to water and other small molecular substances. The venules collect blood from the capillaries and gradually lead into progressively larger veins. The veins function as conducts of transport of blood from the venules back to the heart. Equally important, they serve as the major reservoir of extra blood and sometimes referred to as the capacitance vessel. Capacitance vessels. Because of the pressure in the venous system is very low, the venous walls are thin. Even so, they are muscular enough to contract or expand and thereby serve as a controllable reservoir for the extra blood, either a small or a large amount depending on the needs of the circulation. And volumes of blood in the different parts of the circulation. We just learned that. For instance, about 84% of entire blood volume of the body is in the systemic circulation and 60% is in the heart and lungs. Of 84% in the systemic circulation, approximately 64% is in veins, 13% is in arteries, and 7% is in systemic arterioles and capillaries. The heart contains 7% of blood and pulmonary vessel contain 9%. Most surprising is the low blood volume in the capillaries. It is there, however, that the most important function of the circulation occurs: diffusion of substances back and forth between the blood and function. So cross-sectional areas and velocities of blood flow. If all the systemic vessels of each type were put side by side, their approximate total cross-sectional areas for the average human being would be as follows: like 2.5 iota, small artery 20, arterioles 40, cross-sectional area cm square, like that. Then note particularly that the cross-sectional areas of the veins are much larger than those of the arteries, averaging about 4 times those of the corresponding arteries. This difference explains the large blood storage capacity of the venous system. Large blood storage capacity of the venous system in comparison with the arterial system. Then, because the same volume of blood flow must pass through each segment of the circulation each minute, the velocity of blood flow is inversely proportional to the vascular cross-sectional area, that is E is equal to F by A. Thus, under resting conditions, the velocity average about 33 centimeter per second in the aorta, but is only 1 by 1000 in as rapid in the capillaries, about 0.3 mm per second. However, because the capillaries have a typical length of only 0.3 to 1 mm, the blood remains in the capillaries for only 1 to 3 seconds, which is surprising because all diffusion of nutrient, food substances, and electrolytes that occurs through the capillary walls must be performed in this short time, 1 to 3 seconds in capillaries. Then pressure on the various portions of the circulation. Because the heart pumps blood continuously into the aorta, the mean pressure in the aorta is high, averaging about 100 mmHg. Also, because heart pumping is pulsatile, the arterial pressure alternates between systolic pressure level and a diastolic pressure level of 80 mmHg. Systole and diastole and systole. 80 bar 120. That means 80 is diastole. Okay. As the blood flows through the systemic circulation, its mean pressure falls progressively to about 0 mmHg by the time it reaches the termination of the posterior and inferior vena cava, where they empty into the right atrium of the heart. The pressure in the systemic capillaries varies from as high as 35 mmHg near the arteriolar ends to as low as 10 mmHg near the venous ends. But their average functional pressure in most venular blood is about 17 mmHg, a pressure low enough that little of the plasma leaks through the minute pores of the capillary walls, even though nutrients can diffuse easily through the same pores into the underlying tissue cells. Note that the far right side, representative pressure in the different parts of the pulmonary circulation is present. The pulmonary artery, the pressure is pulsatile, just as in aorta, but the pressure is far less. Pulmonary artery systolic pressure averages about 25 mmHg and a diastolic pressure averages about 8 mmHg with mean pulmonary artery pressure of only 16 mmHg. The mean pulmonary capillary pressure averages only 7 mmHg, yet the total blood flow through the lungs each minute is same as those of the systemic circulation. The low pressure in the pulmonary system are in accord with the needs of the lungs because all that is required is to expose the blood to the pulmonary capillaries to oxygen and other gases in the pulmonary alveoli. Basic principles of circulatory function. Although the details of circulatory function are complex, three basic principles underlie all functions of the system, which are the first one: blood flow to most tissues is controlled according to the tissue need. When tissues are active, they need a greatly increased supply of nutrients and therefore much more blood flow than when they are at rest, occasionally as much as 20 to 30 times in resting level. Yet, the heart normally cannot increase its cardiac output more than 4 to 7 times greater than resting levels. Therefore, it is not possible simply to increase blood flow everywhere in the body when a particular tissue demands increased flow. Instead, the microvessels in each tissue continuously monitor tissue needs, such as availability of microvessels in each tissue continuously monitor tissue needs, such as availability of oxygen and other nutrients, and accumulation of carbon dioxide and other tissue waste products. And these microvessels in turn act directly on the local blood vessels, dilating or constricting them to control local blood flow precisely to that level required for the tissue activity. Also, nerve control of the circulation from the central nervous system and hormones provide additional help in controlling tissue blood flow. Central nervous system play function, hormones also. Okay, the next point. Cardiac output is the sum of the local tissue flows. Cardiac output is the sum of all the local tissue flows. When blood flows through a tissue, it immediately returns by way of its veins to the heart. The heart responds automatically to this increased inflow of blood from pumping it immediately back into the arteries. Thus, the heart acts as an automaton responding to the demands of the tissue. In the heart, however, often needs help in the form of special nerve signals. Heart needs special nerve signals to make it pump the required amount of blood flow. Then arterial pressure regulation is generally independent of either local blood flow control or cardiac output control. Arterial blood regulation is generally independent of either local blood flow control or cardiac output control. The circulatory system is provided with an extensive system for controlling the arterial blood pressure. For instance, if any time pressure falls significantly below the normal level of about 100 mmHg, within seconds a barrage of nerve reflexes elicit a series of circulatory changes to raise the pressure back toward normal. The nerve signal especially increase the force of heart pumping because contraction of the large venous reservoirs to provide more blood to the heart cause constriction in the arterioles in many tissues so that more blood accumulates in the large arteries to increase the arterial pressure. Then over more prolonged periods, hours and days, the kidneys play an additional major role in pressure control, both by secreting pressure controlling hormones by kidney and by regulating blood volume. The next topic, properties of cardiac muscle. The heart is composed of three major types of cardiac muscle: arterial muscle or atrial muscle, ventricular muscle, and the specialized excitatory and conductive muscle fibers, which are the arterial, ventricular, specialized excitatory and conductive muscle fibers. The arterial and ventricular types of muscle contract in much the same way as skeletal muscle, except that the duration of contraction is very much longer. The specialized excitatory and conductive fibers of the heart, however, contract only feebly because they contain few contractile fibrils. Instead, they exhibit either automatic rhythmical electrical discharge in the form of action potentials or conduction of the action potentials through the heart, providing an excitatory system that controls the rhythmic beating of the heart. Once again, the heart is composed of three major types of cardiac muscles: atrial muscle, ventricular muscle, and specialized excitatory and conductive muscle fibers. The atrial and ventricular types of muscles contract in much the same way as skeletal muscle do, except that the duration of contraction is very longer. The specialized excitatory and conductive fibers of the heart, however, contract only feebly because they contain few contractile fibrils. Instead, they exhibit either automatic rhythmical electrical discharge in the form of action potentials or conduction of the action potentials through the heart, providing an excitatory system that controls the rhythmical beating of the heart. Next, anatomical characters of cardiac muscle. Cardiac muscle fibers are arranged in a lattice work with the fibers dividing, recombining, and then spreading again. Cardiac muscle is striated in the same manner as the skeletal muscle. It's striated. Never forget that point. Striated. In the same manner as in skeletal muscle. Further, cardiac muscle has special typical myofibrils that contain actin and myosin filaments almost identical to those found in skeletal muscles. Actin will be there, myosin will be there. Okay, and it's to a greater extent similar to normal muscles. It's straight. Okay, now forget that point. These filaments lie side by side and side during contraction in the same manner as occurs in skeletal muscles. In other ways, however, cardiac muscle is quite different from skeletal muscle, as we shall see. Some other points should say that these cardiac muscles are to a great extent, very different from skeletal muscles. Physiological characteristics of cardiac muscle. Cardiac muscle is excitable, like all other living cells. Cardiac muscle cells are excitable, but that is, they have the ability to respond to a stimulus. The response to a stimulus is a change in the membrane potential, and if the stimulus provided is of sufficient intensity, the biological response, that is contraction, is obtained. Cardiac muscle is excitable. Excitation will be taking place. Excitable. Factors that affect the excitability of heart is referred to as bathymotropic factors. Bathymotropic factors. I'm going to write that here. Bathymotropic factors. Bathymotropic factors. These factors that increase excitability are called positively bathymotropic. Once again, factors that affect the excitability of a heart are referred to as bathymotropic factors. Those factors that increase excitability are called positively bathymotropic, while those that reduce excitability are called negatively bathymotropic. Then autorhythmicity. Earlier we learned excitable function, and bathymotropic factors. Next, autorhythmicity. Cardiac muscle is self-excitatory. Although this property can be modulated by nerves, hormones, and drugs. The ability of the heart to generate its own rhythm is discussed later, so we'll be learning that. Then cardiac muscle is contracted. Autorhythmicity, then contracted. This is self-explanatory. However, cardiac muscle contraction differs in important ways from skeletal muscle contraction, and these are discussed later. So, inotropy refers to the force of muscle contraction. Inotropy. Okay. Factors that increase the force of contraction are called positively inotropic, while positively inotropic. Earlier we learned bathymotropic in the sense excitability factors, excitability increasing factor, positively bathymotropic. These are about inotropic in the sense they just contract this. Then cardiac muscle conducts electrical impulses. We know that. The presence of intercalated discs is especially important. Intercalated discs, never forget to write that. Important in allowing cardiac muscles to conduct electrical impulses. However, the speed with which impulses are conducted in cardiac muscle is relatively slow. The heart has in place a specialized conducting system. Cardiac muscle is a syncytium. This point you might have heard, but it's a very important point. The dark areas crossing the cardiac muscle fibers are called intercalated discs. And some intercalated discs we just mentioned earlier. At each intercalated disc, the cell membrane fuse with each other in such a way to form permeable communicating junction or gap junctions that allow rapid diffusion of ions. Therefore, from a functional point of view, ions move with ease in the intracellular fluid so that action potentials travel easily from one cardiac muscle cell to the next past the intercalated disc. Thus, cardiac muscle is a syncytium of many heart muscle cells. In which the cardiac cells are so interconnected that when one cell becomes excited, the action potential spreads to all of them. Like that. Once again, just have a quick recap. Cardiac muscle is excitable. Cardiac muscle have autorhythmicity. Cardiac muscle is contractile. Cardiac muscle conducts electrical impulses. Bathmotropic factors affect the excitability, and inotropic factors cause contraction, excitable contraction. Then cardiac muscle conduct electrical impulses, cardiac muscle is a sensation. That point we are going to explain also. So we just learned sensation of many heart muscle cells create a cardiac muscle. The heart is actually composed of two sensations: the atrial sensation, which constitute the walls of the two atria, and the ventricular sensation. Atrial and ventricular sensation, which constitute the walls of two ventricles. The atria are separated from ventricles by fibrous tissue that surrounds the atrioventricular valvular openings between the atria and ventricle. Normally, potentials are not conducted from atrial sensation into the ventricular sensation directly through this fibrous tissue. Instead, they are conducted only by way of a specialized conductive tissue called AV bundle. Specialized conductive tissue called AV bundle. AV bundle. AV bundle. Okay. This division of the muscle of the heart into two functional sensations allows the atria to contract in short time ahead of ventricular contraction. This allows the blood in the atria to empty into ventricles before the ventricles begin contracting. Otherwise, the whole system will be collapsing. So first, the atrium will be contracting. After that, ventricles will be contracting. The all or none low in the heart applies to the entire sensation. All or none low. For skeletal muscle, a stimulus that is of threshold or greater than threshold intensity applies to a motor nerve fiber will result in the contraction of all the muscle fibers applied by the nerve. That is the entire motor unit. In the case of the heart, because cardiac muscle behaves like a sensation, the threshold or greater than threshold stimulus applies to any part of the sensation will result in the contraction of the entire sensation. That's a very unique point. That means threshold or greater than threshold stimulus applied to any part of this sensation will result in the contraction of the entire sensation, because that acts as a single unit. Then the action potential in cardiac muscle is prolonged and has a long refractory period. Long refractory period. The action potential recorded in a ventricular muscle fiber is important. So after the initial spike, the membrane remains depolarized for about 0.2 seconds, exhibiting a plateau followed at the end of the plateau by abrupt repolarization. The presence of this plateau in the action potential causes ventricular contraction which lasts as much as 15 times as long in cardiac muscle as a skeletal muscle. One of the characteristics of cardiac muscle, like all excitable tissue, is that it is refractory to re-stimulation during the action potential. Therefore, the refractory period of the heart is in the interval of time as shown to the left. So, during which a normal cardiac muscle impulse cannot re-excite an already exciting area of cardiac muscle. The normal refractory period of the ventricle is 0.25 to 0.30 seconds, which is about the duration of the prolonged plateau action potential. There is an additional relative refractory period of about 0.05 seconds, during which the muscle is more difficult to excite than normal, but nevertheless can be excited by a very strong excitatory signal, as demonstrated by the early premature contraction of the second example. The refractory period of arterial muscle is much shorter than that of the ventricles, almost about 0.15 second for the atria compared with 0.25 to 0.30 second for the ventricles. The long refractory period is one of the factors why cardiac muscles does not fatigue. Refractory period is high. Long refractory period. Long refractory period is one of the reasons why cardiac muscle does not fatigue. Then cardiac muscle require calcium in the extracellular fluid to contract. As is true for skeletal muscle, when an action potential passes away the cardiac muscle membrane, the action potential spreads to the interior of the cardiac muscle fiber along the membrane of the transverse tubule. The T-tubule action potential in turn act on the membranes of the longitudinal sarcoplasmic tubules to cause release of calcium ions into the muscle sarcoplasm from the sarcoplasmic reticulum. In another few thousands of seconds, these calcium ions diffuse into the myofibrils and catalyze the chemical reactions that promote sliding of an actin and myosin filaments along one another, which is produced by the muscle contraction. This far, this mechanism of excitation contraction coupling is same as that of skeletal muscle, but there is a second effect that is quite different. In addition to the calcium ions that are released into the sarcoplasm from the cisternae of the sarcoplasmic reticulum, calcium ions also diffuse into the sarcoplasm from the T tubules themselves at the time of action potential, which opens voltage dependent calcium channels in the membrane of the T tubule. We just learned some another points like all or none low. The action potential in cardiac muscle is prolonged and with a long refractory period. Long refractory period will be there. Cardiac muscle require calcium in the extracellular fluid to contract. Calcium is a requirement. Calcium entering the cell then activates calcium release channels, also called ryanodine receptor channels. Ryanodine receptor channels in the sarcoplasmic reticulum membrane, triggering the release of calcium into the sarcoplasm. Calcium ions in the sarcoplasm then interact with the troponin to initiate cross bridge formation and contraction by the same basic mechanism as the skeletal muscle. Without the calcium from the T tubule, the strength of the cardiac muscle contraction would be reduced considerably because of the sarcoplasmic reticulum of cardiac muscle is well developed than that of skeletal muscle and does not store enough calcium. To provide full contraction. The T tubules of cardiac muscle, however, have a diameter of 5 times greater than that of a skeletal muscle tubule, which means 25 times greater volume. Also, inside the T tubule is a large quantity of mucopolysaccharides that are electronegatively charged. Mucopolysaccharides are there, which are electronegatively charged and bind an abundant store of calcium ions, keeping them available for diffusion to the interior of the cardiac muscle fiber when a T tubule action potential appears. The strength of the contraction of the cardiac muscle depends on a great extent on the contraction of calcium ions in the extracellular fluids. In fact, the heart placed at a calcium-free solution will quickly stop beating. The reason for this response is that opening of the T tubules pass directly through the cardiac muscle cell membrane into the extracellular spaces surrounding the cells, allowing the same extracellular fluid that is in the cardiac muscle interstitium to percolate through the T tubule. Consequently, the quantity of calcium ions in the T tubule system, the availability of calcium ions to cause cardiac muscle contraction, depends to a greater extent on the extracellular fluid calcium ion concentration. So, sarcoplasmic reticulum is associated with this. And cardiac muscle contraction is dependent on the initial length of the muscle fiber, Frank-Starling law of the heart, and the concepts of preload and afterload. The degree of tension on the muscle when it begins to contract is called the preload. Degree of tension before contraction is called the preload, and when the load against which the muscle exerts its contraction force is called the afterload. The load against which the muscle exerts its contractile force is called afterload. For cardiac contraction, the preload is usually considered to be the end diastolic pressure when the ventricle has become filled. The greater the end diastolic volume, ventricular filling, and the greater the corresponding end diastolic pressure, the greater is the strength of the cardiac muscle. This increased initial length to the muscle results in a greater force of contraction within physiological limits and is referred to as a Frank-Starling law of the heart. The relationship of length to contractibility is modified by the pressure of and presence of inotropic factors. For instance, intense sympathetic stimulation can increase the heart rate in young humans for the normal rate of 70 beats per minute up to 180 to 200, rarely even 250 beats per minute. Sympathetic stimulation also increases the force of heart contraction to as much as double the normal volume. Similarly, increasing the volume of blood pumped and increasing the ejection pressure. The sympathetic stimulation often can increase the maximum cardiac output as much as twofold to threefold in addition to the increased output caused by the Frank stalling mechanism already discussed. Once again, we are going to learn that mechanism. For cardiac contraction, the preload is something else, which is the term which we just mentioned. The degree of tension on the muscle when it begins to contract is called the preload, while the load against which the muscle exerts its contractile force is called afterload. Preload and afterload. For cardiac contraction, the preload is usually considered to be the end diastolic pressure when the ventricle has become filled. The greater the end diastolic volume or ventricular filling, and the greater the corresponding end diastolic pressure, the greater is the stretch of the cardiac muscle. It increased initial length of muscle results in a greater force of contraction within physiological limits, and this is referred to as Frank-Starling law of heart. Increased initial length of the muscle results in a greater force of contraction within physiological limits. Frank-Starling law of heart. Frank-Starling law of heart. Okay. Conversely, inhibition of the sympathetic nerves to the heart can decrease cardiac pumping to a moderate extent in the following way. Under normal conditions, the sympathetic nerve fiber to the heart discharge continuously at a low rate that maintains pumping of about 30% of above that with no sympathetic stimulation. Therefore, when the activity of the sympathetic nerve system is depressed below normal, this decreases both heart rate and strength of muscular muscle contraction, thereby decreasing the level of cardiac pumping as much as 30% below normal. The afterload of the ventricle is the pressure in the aorta leading from ventricle. This corresponds to the systolic pressure described in the phase 2 curve of volume pressure diagram. Sometimes the afterload is loosely considered to be the resistance in the circulation rather than the pressure. The importance of the concept of preload and afterload is that in many abnormal functional states of the heart or circulation, the pressure during filling of the ventricle or the preload and arterial pressure against which the ventricle must contract, the afterload for both are altered from normal to severe degree. Next, cardiac muscle does not fatigue. It is truly amazing that the heart continues to beat in a healthy individual with such regularity in entire lifespan of an individual without getting tired. Even the most well-trained skeletal muscle in the most elite of the athletes cannot achieve this. There are several reasons why cardiac muscles does not get fatigued. Cardiac muscle has a long refractory period we mentioned earlier. This cardiac muscle cannot be tetanized, while tetany allows for the generation of greater force. Fatigue also sets in much faster. Cardiac muscle has a rich blood supply. Rich blood supply will be there. So in context, cardiac muscle has a capillary density that is about 4 times greater than skeletal muscles. Capillary density is also high. Capillary density is higher. Cardiac muscle undergoes aerobic metabolism. Aerobic metabolism, not anaerobic, aerobic. Thus, the accumulation of lactic acid, an important factor in the development of fatigue in anaerobically exercising muscles, play no role in cardiac muscle. This one is a very important point because we know lactic acid accumulation occur in case of anaerobically metabolizing muscle fibers. So this is not happening in case of cardiac muscle because the process is aerobic metabolism, not anaerobic, aerobic metabolism. Aerobic metabolism. Okay, so cardiac muscle is rich in mitochondria. Mitochondria is also high concentration. Then capillary density is also high. Then mitochondria account for almost 1/3 of the cardiac muscle by weight and are best appreciated by an examination of the histology of cardiac muscle. A great amount of mitochondria is there. So first, let's have a quick recap, very quick. Properties of cardiac muscles. Which are the major properties? Cardiac muscle fibers are arranged in a lattice work with the fibers dividing, recombining and then spreading again. A lattice is formed. They are striated, contain actin and myosin filaments just as found in the skeletal muscles. They are of many types like atrial muscle, ventricular muscle and specialized excitatory and conductive muscle fibers. Then cardiac muscle is excitable, bathmotropic factors initiate that excitability. Then autorhythmicity is there, self-excitatory property, autorhythmicity, modulated by nerves, hormones and drugs. Then cardiac muscle is contractile, autorhythmicity is there, excitable, pathomotropic factors, contractile function is also there. So inotropic factors influence that contractile property. Then cardiac muscle conducts electrical impulses and intercalated discs are there. Then cardiac muscle is a syncytium of intercalated discs are also present. Syncytium of many heart muscle cells and arterial syncytium and ventricular syncytium, two types. Then AV bundle, importance of AV bundle, the all or none low. A threshold or greater than threshold stimulus applied to any part of a syncytium will result in the conduction of the entire syncytium. Okay, then a long refractory period is their peculiarity. Cardiac muscle require calcium in the extracellular fluid to contract. So importance of ryanodine receptor channels, calcium release channels, ryanodine release channels, ryanodine, RYANODINE, ryanodine receptor channels, calcium releasing channels. They are very important. T tubules, just write about T tubules, does not store enough calcium. Skeletal muscle store calcium, but cardiac muscle doesn't store that much calcium. Then mucopolysaccharides are present. Then heart placed in a calcium-free solution will quickly stop beating. Then cardiac muscle contraction is depend on the initial length of the muscle fiber. Cardiac muscle contraction is depend on the initial length of the muscle fiber. That explains the Frank-Starling law of heart and concepts of preload and afterload. Frank-Starling law of the heart. Then some other aspects related to cardiac muscle does not fatigue. Why? Because cardiac muscle has a strong refractory period, first point. Rich blood supply, capillary density is high, aerobic metabolism will be taking place, so no lactic acid accumulation. And cardiac muscle is rich in mitochondria. Once again, these points are very important. Cardiac muscle does not fatigue. Why? Because it has long refractory period. Rich blood supply, capillary density is high, aerobic metabolism will be taking place, so no accumulation of lactic acid, unlike muscle which contain lactic acid accumulation due to anaerobic metabolism. Then rich in mitochondria. These are major points related to cardiac muscle properties. Next topic is cardiac cycle. Heart is actually two separate pumps. The right heart that pumps blood through the lungs, right heart, and a left heart that pumps blood through the systemic circulation. Right through lungs and left through systemic circulation that provides blood flow to the other organs and tissues of the body. Always remember right lung, like that, right lung. Okay, so in turn each of this heart is a pulsatile two chamber pump composed of an atrium and ventricle. We know that the ventricles supply the main pumping force that propels the blood either through the pulmonary circulation by the right ventricle or through the systemic circulation by the left ventricle. What is happening? The ventricles supply the main pumping force that propels the blood either through the pulmonary circulation by the right ventricle or through the systemic circulation by the left ventricle. The cardiac events that occur from the beginning of one heartbeat to the beginning of the next is called cardiac cycle. Cardiac events are taking place which occur from the beginning of one heartbeat to the beginning of the next are called cardiac cycle. Very simple idea. This cycle is initiated by spontaneous generation of an action potential in the sinus node. Now we are going to run some complex topics. Each cycle is initiated by the spontaneous generation of an action potential in the sinus node. An action potential travels from here rapidly through both atria and even through the atrioventricular bundle, AV bundle, into the ventricles. Through which one? AV bundle, sinus node, generated by sinus node. Sinus node. Okay. Because of the special arrangement of the conducting system from the atria into the ventricle, there is a delay of more than 0.1 second during the passage of the cardiac impulse from the atria into the ventricle. This delay allows the atria to contract ahead of the ventricular contraction. We mentioned this point earlier, thereby pumping blood into the ventricles before the strong ventricular contraction begins. Thus, the atria acts as a primer pumps for ventricles. Primer in the sense earlier, beating earlier. And the ventricles in turn provide the major source of power for moving blood through the body's vascular system. Next major topic, diastole and systole. The cardiac cycle consists of a period of relaxation called diastole, relaxed diastole, during which the heart fills with blood, followed by a period of contraction called systole. Contractile period, systole, relaxation period, diastole, but during that diastole, heart is filled with blood. So the total duration of the cardiac cycle, including systole and diastole, is the reciprocal of the heart rate. For example, if heart rate is 72 beats per minute, the duration of one cardiac cycle is 0.833 second per beat. Approximately 0.833 second per beat. Vigor's diagram show Vigor, WIGGERS diagram show the different events occurring in the cardiac cycle for the left side of the heart. Top three curves show the pressure change in the aorta, left ventricle and left atrium. The fourth curve depicts the change in left ventricular volume. If you need to know the structure, just Google Vigor's diagram about this diastole and systole cardiac cycle. And just the fourth curve depicts the changes in left ventricular volume. The fifth depicts the electrocardiogram, and the sixth depicts a phonocardiogram, which is a recording of the sound produced by the heart. Once again, the top three curves show the pressure changes in the aorta, left ventricle and left atrium. The fourth curve depicts the changes in left ventricular volume, and the fifth depicts the electrocardiogram, and the sixth depicts the phonocardiogram, which is a recording of the sounds produced by the heart, mainly by the heart valves as it pumps. It is especially important that the reader study in detail this figure and understand the causes of all the events shown. In order to simplify things, this diagram is broken up to different kinds. First point, increasing heart rate decreases the duration of cardiac cycle. When heart rate increases, the duration of each cardiac cycle decreases, including the contraction and relaxation phases. The duration of the action potential and the period of contraction, systole, also decrease, but not as a great a percentage as does the relaxation phase, diastole. As a normal heart rate of 72 beats per minute, systole comprises about 0.4 of the entire cardiac cycle. At 3 times the normal heart rate, systole is about 0.65 of the entire cardiac cycle. This means that the heart beating at a very fast rate does not remain relaxed long enough to allow complete filling of the cardiac chambers before the next contraction. Earlier we learned two phases are there, systole and diastole. Systole in the sense contractile phase, diastole is a relaxation phase. So when the heart is beating so fast, the systole phase won't be lower. Systole phase is just like that, but the diastole phase will be reduced. Okay, so mechanical events in the heart. Next topic, mechanical events in the heart. The ventricle fills with blood during diastole. During ventricular systole, large amount of blood accumulates in the right and left atria because of the closed AV bundles. Therefore, as soon as systole is over and the ventricular pressure fall again to the low diastolic value, a moderately increased pressure that are developed in the atria during ventricular systole immediately push the AV valves open and allow blood to flow rapidly into the ventricles. So, ventricular volume curve increase. Left one. So this period is called period of rapid filling of the ventricles. The period of rapid filling lasts about the first third of diastole. During the middle third of the diastole, only a small amount of blood normally flows into the ventricle. This is blood that continues to empty into the atria from the veins and passes through the atria directly into the ventricles. During the last third of diastole, the atria contract and given an additional thrust to the inflow of blood into the ventricles. This mechanism accounts for 20% of filling of the ventricles during each heart cycle. Since about 80% of the blood flows directly through the atria into the ventricles even before the atria contract, the atria simply functions as a primer pump that increases the ventricular pumping effectiveness. However, the heart can continue to operate under most conditions even without this extra 20% effectiveness because it normally has the capacity to pump 300 to 400 more blood than it is required for the resting period. Therefore, when the atria fail to function, the difference is unlikely to be noticed unless a person exercises. Then acute signs of heart failure occasionally develop, especially shortness of breath. So we must exercise at least to find out if our atria is working properly or not. Otherwise, we are not going to know that. If acute signs of heart failure is there, that will emerge when we exercise. That will indicate the atrial dysfunction. An outflow of blood from the ventricles during systole. Period of isometric contraction. Immediately after ventricular contraction begins, the ventricular pressure rises abruptly and causing the AV valves to close when the additional 0.02 to 0.03 second is required for the ventricles to build up sufficient pressure to push the semilunar aortic or pulmonary valves open against the pressures in the aorta and pulmonary artery. Therefore, during this period, contraction is occurring in the ventricles but no emptying occurs. This period is called the period of isovolumic or isometric contraction. Period of isovolumic or isometric contraction, meaning that the cardiac muscle tension is increasing but little or no shortening of the muscle fibers is occurring. Then period of ejection, when the left ventricular pressure rises slightly above 80 mmHg and the right ventricular pressure rises slightly above 8 mmHg, the ventricular pressure pushes the semilunar valves open. Immediately, blood begins to pour out of the ventricles. Approximately 60% of the blood in the ventricle at the end of the diastole is ejected during systole. 70% of this portion flows out during the first third of the ejection period, with the remaining 30% emptying during the next 2/3. So therefore, the first third is called the period of rapid ejection and the last 2/3 are called the period of slow ejection of blood. So posterior period of isovolumic or isometric relaxation. At the end of systole, ventricular relaxation begins suddenly, allowing both the right and left intraventricular pressures to decrease rapidly. The elevated pressure in the distended large arteries that have just been filled with blood from the contracted ventricles immediately push blood back towards the ventricles, which snaps the aortic and pulmonary valves closed. This brief period during which there is a backflow of blood associated with the closure of the aortic and pulmonary valves is called protodiastole. For another, what is that? Protodiastole period. For another 0.03 to 0.06 second, the ventricular muscle continues to relax even though the ventricular volume does not change, giving rise to the period of isovolumic or isometric relaxation. During this period, the intraventricular pressure rapidly decreases back to their low diastolic levels, and the AV valve opened to begin a new cycle of ventricular pumping. The ventricular pressure volume changes depicting separately. So that's the phase of cardiac cycle. That's the phase of the cardiac cycle as follows. A rapid filling of the ventricles will be taking place. A slow filling of the ventricles. Atrial systole. Isovolumetric contraction. Isovolumetric contraction. Rapid ejection. Slow ejection. Photodiastole. Isovolumic relaxation. Then electrical events in the cardiac cycle. The electrocardiogram is very important with PQRS and T waves. They are electrical voltages generated by the heart and recorded by the electrocardiograph from the surface of the body. P wave is caused by the speed of depolarization through the atria and is followed by atrial contraction, which causes a slight rise in the atrial pressure curve immediately after the electrocardiogram is graphic P wave. So spread of depolarization indicate P wave. Which depolarization? Atrial depolarization. Although 0.16 second after the onset of P wave, the QRS waves appear as a result of electrical depolarization of the ventricles, which initiate contraction of the ventricle and cause the ventricular pressure to begin rising, as also shown in a figure which you need to Google. So therefore the QRS complex begins slightly before the onset of ventricular systole. Finally, the ventricular T wave represents the stage of a repolarization of the ventricle when the ventricular muscle fibers begin to relax. Therefore, the T wave occurs slightly before the end of ventricular contraction. These changes are depicted. So, pressure changes in the atria A, C, and V waves. In the atrial pressure curve, three minor pressure elevations called A, C, and V atrial pressure waves are shown. The A wave is caused by an atrial contraction. Ordinarily, the right atrial pressure increases 4 to 6 mmHg during atrial contraction, and the left atrial pressure increases about 7 to 8 mmHg. The C wave occurs when the ventricles begin to contract. It is caused partly by slightly backward flow of blood into the atria at the onset of ventricular contraction, but mainly by bulging of the AV valves backward toward the atria because of increasing pressure in the ventricles. The V wave occurs towards the end of the ventricular contraction. It results from the slow flow of blood into the atria from the veins, while the AV valves are closed during ventricular contraction. Then, when ventricular contraction is over, the AV valves open, allowing the stored atrial blood to flow rapidly into the ventricles and causing the V wave to disappear. This right atrial pressure changes that are also reflected in the jugular venous pressure traces are very important. Then, aortic pressure curve. When the left ventricle contracts, the ventricular pressure increases rapidly until the aortic valve opens. Then, after the valve pressure opens, the pressure in the ventricle rises much less rapidly, because blood immediately flows out of the ventricle into the aorta and then into the systolic distribution of arteries. The entry of blood into the arteries during systole causes the walls of these arteries to stretch and the pressure to increase to about 120 mmHg. Next, at the end of the systole, after the left ventricle stops ejecting blood and the aortic valve closes, the elastic walls of arteries maintain a high pressure in the arteries even during diastole. Incisura occurs in the aortic pressure curve when the aortic valve closes. This is caused by a short period of backward flow of blood or protodiastole immediately before closure of the valve, followed by sudden cessation of the backflow. After the aortic valve has closed, the pressure in the aorta decreases slowly throughout diastole because of the blood stored in the distended elastic fibers of arteries follows continuously through the peripheral vessels back to the veins. Before the ventricular contraction again, aortic pressure usually has fallen to about 80 mmHg diastolic pressure, which is 2/3 of the maximum pressure of 120 mmHg diastolic pressure that occur in the aorta during ventricular contraction. The pressure curve in the right ventricular and pulmonary artery is similar to those of the aorta, except that the pressure is only about 1/6 as great as discussed. Heart sounds during cardiac cycle. When listening to heart with a stethoscope, one does not hear the opening of the valves because it is a relatively slow process that normally makes no noise. However, when the valves close, the veins of the valves and the surrounding fluids vibrate under the influence of sudden pressure change. Why this is happening? Veins of valves and the surrounding fluids. Veins of valves and surrounding fluids vibrate under the influence of sudden pressure change. So that sound travels in all directions of the chest. Fluid vibration and vein valves. When the ventricles contract, one first hears a sound caused by the closure of the AV valve. This vibration, which is slow, the AV valve will be closing first, and relatively long lasting, and it is called the first heart sound. When the aortic and pulmonary valves close at the end of the systole, one hears a rapid snap because these valves close rapidly and the surrounding vibrates for a short period. The sound is called the second heart sound. So first heart sound is nothing else but the closure of the AV valve. That is the first heart sound. Then second heart sound is the aortic and pulmonary valves close at the end of the systole. Okay. Then the third heart sound relates to the turbulence of blood in the rapid filling phase, and the fourth heart sound related to the turbulence of blood during atrial systole, which may not always be heard. The location of this heart sounds in relation to cardiac cycle is important. The graphical analysis of ventricular pumping, which to a great extent require a graph. So a diagram which is especially useful in explaining the pumping mechanics of left ventricle. The most important components of diagram and the two curves located. Diastolic pressure and systolic pressure. These curves are volume pressure curves. The diastolic pressure curve is determined by filling the heart with progressively greater volumes of blood and then measuring the diastolic pressure immediately before ventricular contraction occurs, which is the end diastolic pressure of the ventricle. The diastolic pressure curve is determined by recording the systolic pressure achieved during ventricular contraction at each volume of filling. Until the volume of non-contracting ventricle rises above about 150 ml, the diastolic pressure does not increase greatly. Therefore, up to this volume, blood can flow easily into the ventricle from the atrium. Above 150 ml, the ventricular diastolic pressure increased rapidly, partly because of the fibrous tissue in the heart that will scratch no more, and partly because of the pericardium that surrounds the heart become filled nearly to its limit. During ventricular contraction, the systolic pressure increases even at low ventricular volumes and reaches a maximum at a ventricular volume of 150 to 170 ml. Then the volume increases still further. The systolic pressure actually decreases under such conditions and demonstrated. Then this great volumes, the actin and myosin fibers of cardiac muscle fibers are pulled apart far enough to the length of each cardiac fiber contraction becomes they are less optimal. Volume pressure diagram during the cardiac cycle, cardiac output, volume pressure diagram. Period of filling, phase 1. Phase 1 is the volume pressure diagram that begins at a ventricular volume of about 50 ml and a diastolic pressure of 2 to 3 mmHg. So there diastolic pressure is low and systolic volume is there. Then period of isovolumic contraction, then period of ejection, period of isovolumic relaxation, then etc. will be happening. Then oxygen utilization by the heart. Heart muscle, like skeletal muscle, uses chemical energy to provide the energy for contraction. Approximately 70 to 90% of this energy is normally derived by oxidative metabolism of fatty acids, with about 10 to 30% coming from other nutrients, especially lactate and glucose. Therefore, the rate of oxygen consumption by the heart is an excellent measure of the chemical energy liberated while the heart performs its work. Experimental studies have shown that oxygen consumption of heart and the chemical energy expanded during contraction is directly related to the total area. So this portion is called potential energy. The potential energy represents additional work that could be accomplished by contraction of the ventricle if the ventricle should completely empty all the blood in its chamber with its contraction. Oxygen consumption has also been shown to be nearly proportional to the tension that occurs in the heart muscle during contraction multiplied by the duration of time of that contraction persists, given the tension time index. Because tension is high when systolic pressure is high, correspondingly more oxygen is used. Also, much more chemical energy is expanded even at normal systolic pressure when the ventricle is abnormally dilated, because heart muscle tension during contraction is proportional to the pressure times the diameter of the ventricle. This becomes especially important in heart failure when the heart ventricle is dilated, and paradoxically the amount of chemical energy required for a given amount of work output is greater than normal when the heart is already failing. Then efficiency of cardiac contraction. During heart muscle contraction, most of the expanded chemical energy is converted into heat, and a much smaller portion is converted to work output. The ratio of work output in the total chemical energy expenditure is called the efficiency of cardiac contraction, or simply efficiency of the heart. Maximum efficiency of normal heart rate is between 20 and 25%. In persons with heart failure, the efficiency can decrease to as low as 5 to 10%. Most of the expended chemical energy is converted into heat, and much smaller amount is converted into work output. The ratio of work output to total chemical energy expenditure is called efficiency.